Enzymes as diagnostics. We all know enzymes are biocatalysts because they have been widely used in industrial processes such as food processing, beer fermentation, laundry detergents, pickling purposes, and as well as to control or accelerate the catalytic reactions in order to quickly and precisely obtain various valuable end products. So enzymes are generally biocatalysts, but they are highly used as diagnostic tools in medical field also. Let us explore it. These are the topics that are described in this lecture, Enzymes as Diagnostics. The first one is introduction as how are enzymes utilized in diagnostics, and the second one, enzymes in diagnostics of diseases, especially we are going to deal with the liver diseases as well as cancer. And the third subtopic, enzymatic biosensors as diagnostic tools. So not only in the diseases, it is separately used as a tools also. And the fourth one, allosteric enzyme-based biosensors, a modification of an enzyme called allosteric enzyme and how they are utilized in biosensors. And the last category of this lecture, commercially available enzyme-based biosensors, and talks about the recent technological innovations about enzyme-based biosensors that are commercially available in the market. Introduction. Enzymes that are used for the detection or diagnosis or prognosis of disease conditions are called diagnostic enzymes. Moreover, due to their substrate specificity and quantitative activity in the presence of other proteins, enzymes are preferred in diagnosis and therefore can be used as a diagnostic tool for disease detection. There are specific characteristics that makes enzymes as valuable tools for the clinical diagnosis of various diseases. The first one is high specificity. Enzymes are highly specific towards the substrate. It doesn't have any non-targeted activity. So the first characteristic is high specificity. And the second one is sensitivity. It is related to with its Km value. Depending on the concentration of the substrate, enzymes are highly sensitive to the substrates, and it performs a kind of a key and lock model in which it binds alone with the substrate at specific site known as binding site. And the third one is rapid response. Once bound with the substrate, it performs a reaction much faster than the ordinary reaction that is without these enzymes. So enzymes with its own characteristic as biochemical catalyst, which means which lessens the activation energy of the reaction. So it gives a rapid response. And the fourth one is ease of self-testing. So enzymes are utilized here as biosensors. So it can be used for self-testing other than going for a laboratory or a clinic for performing laboratory tests. And the fourth one is portability. Now enzymes as biosensors are available as compact devices in commercial markets, so that can be checked or diagnosed on its own under proper diagnosis by the doctor. And there are many more such applications which can be dealt in this topic, and we are going to see all those things. The most successful commercialized enzymatic biosensor is the glucose biosensor. It is used for the detection of glucose levels in blood samples during diabetic conditions. So, enzymes as well as enzyme-based biosensors both are used as diagnostic tools. Enzymes are also used as diagnostic tools, and enzyme-based biosensors are diagnostic tools. So they are both powerful tools for the detection of other diseases such as cancer, heart failure, epilepsy, and so forth, which are promising in the near future. Enzyme-based biosensors are enzymes as its own. They are not only used in diagnostic purposes, they are also used in prognosis also. The difference between diagnosis and prognosis. Diagnosis depicts the condition, the process of identifying the illness along with its causes and symptoms, which is the occurred event. But regarding the prognosis, it is the process of predicting how soon the illness will progress. So for both the purposes, enzymes are used as diagnostic tools as well as prognostic tool. And the last category, why enzymes are used as diagnostics. Normally it is not only, it has both quantitative properties as well as qualitative properties. Quantitative properties, it will ensure you how much products are formed from a substrate. Here substrate indicates the biomarker that is present in them, especially for a disease or any other condition. So it not only quantitates the level of the product formed, but it also reveals the qualitative analysis of those products. So enzymes are used both for quantification as well as qualitative analysis. Enzymes in diagnosis of diseases. First of all, we will see in liver diseases, but before going into that, the diagnosis of the serum level of certain enzymes has been used as an indicator of cellular damage that results in the release of intracellular components into the bloodstream. So, whenever there is a cellular damage, there are components that are present into the cell or released from it. So, these components are acted by several enzymes, and these act as a biomarker for those rupture of that cell, which can be indicated from these enzymes, which are used as in diagnostics. So if you go to a physician, and for example if he is a neurologist, and if he diagnoses his patient to undergo a neurological enzyme assay, it has a purpose. The purpose is to ascertain or to know whether or not there is brain damage. For the diagnosis of diseases, numerous enzymes have been used as biosensors. Commonly assayed enzymes for the diagnosis of various diseases are alkaline phosphatase, creatine kinase, aminotransferases, including alanine aminotransferase and aspartate aminotransferase, dehydrogenases, sorbitol dehydrogenase and lactate dehydrogenase, cholinesterase, cyclooxygenase, tartarate resistant acid phosphatase, and so on. Many other enzymes are also involved in human as well as veterinary medicine for the clinical diagnosis of different diseases. First, we will see in liver diseases. Describing about liver, it is the largest internal organ, and I have told you that it is a traffic place of human body. It plays an important role in many body functions. It includes detoxification of blood, cholesterol, glucose, iron metabolism, and so on. So various conditions affect the normal functioning of the liver. So that could result into hepatitis, Epstein-Barr virus infection, fatty liver, excessive intake of alcohol, that is cirrhosis, and overtake of drugs such as acetaminophen, and so on. During these conditions, that is abnormal conditions, various enzymes are released into the bloodstream, and quantification and qualitative analysis of these catalysts could lead to the detection of that particular liver disease. In liver, the first category of enzyme, aminotransferases. These are prominent enzyme that is present in the liver, that is secreted from the liver. They are also known as transaminases. They are a group of enzymes that carry the interconversion of amino acids and oxo acids. Example, oxaloacetate, alpha-ketoglutarate, those are called oxo acids. It involves the transfer of amino groups from amino acids as well as oxo acids in a reversible manner. And in this category, there are specialized enzymes known as alanine aminotransferase. Alanine aminotransferase involves transfer of amino group from alanine as well as from oxo acids, which is shortly known as ALT. It was formerly known as glutamate pyruvate aminotransferase. And aspartate aminotransferase, that is the second category. It is also known as glutamate oxaloacetate aminotransferase. So the name depicts that for ALT, the glutamate is formed from pyruvate and so on. Pyruvate is formed from glutamate in a reversible reaction. In the same way for aspartate aminotransferase, so aspartate is formed from glutarate. So these are two clinically important enzymes that comes under the classification of aminotransferases. So the first category, alanine aminotransferase, where it is utilized as diagnostic tool. So in liver disorders, the most prevalent health problems are hepatitis and hepatocirrhosis, which are caused by different factors, including excessive alcohol intake, imbalanced diet, lack of exercise, insufficient sleep, and overconsumption of high-calorie food. So for this, in clinical diagnosis, the level of ALT acts as an important indicator of potential liver disease. Inside the cell cytoplasm, ALT, that is alanine aminotransferase, catalyzes the reversible transamination of L-alanine and 2-oxoglutarate into pyruvate and glutamate, respectively. In a normal healthy human adult, the ALT concentration ranges from 5 to 35 units per liter, and its concentration above this range indicates a damaged or diseased liver. That includes even heart and muscles also, which is damaged as well as diseased. Alanine ALT concentration was determined by spectrophotometric, calorimetric, or chromatographic methods. Recently, simple and low-cost paper-based analytical devices known as PADs, that is paper-based analytical devices, are used for ALT determination, which have attracted the significant interest of various researchers. And the second important enzyme of aminotransferases are aspartate aminotransferase. The serum level of AST, shortly known as AST for aspartate aminotransferase, helps people to diagnose damaged body organs, especially the heart and liver. So this AST catalyzes the transamination of L-aspartate and 2-oxoglutarate into oxaloacetate and glutamate. So the ranges for a healthy human adult are, it has a concentration of around 5 to 40 units per liter. But in case of severe damage of liver, the AST level rises 10 to 20 times higher than the normal range. It is also found in the red blood cells, muscle tissue, and other organs, including the kidney and pancreas. So it can be used in combination with other enzymes to monitor myocardial, hepatic parenchymal, and muscle diseases in humans as well as animals. Moreover, to screen the liver fibrosis in chronic hepatitis B, the AST to platelet ratio index, so that is aspartate aminotransferase and platelet ratio index, could be used as a marker when elastography is not possible. So elastography is analyzed for this platelet ratio. So that assay, if that assay could not be performed, this alternative assay can be performed, that is called AST to platelet ratio. So this gives a ratio of these two indices about that platelet count. After these aminotransferases, which is the prominent enzyme in liver diseases, the other important enzymes are ALP and GGT. ALP, which is nothing but alkaline phosphatase, it hydrolyzes the phosphate ester bonds in an alkaline environment. The rise in the level of serum ALP is a useful marker of liver disease, particularly cholestatic diseases in which bile ducts are being blocked, as in the case of obstructive jaundice. And the last one regarding liver diseases, GGT, gamma glutamyl transferase. It is present in the cell membranes of almost all human cells and helps in the transport of amino acids from one peptide to another. Therefore, GGT is sometimes also referred to as a transpeptidase. It is found abundantly in the kidney, liver, pancreas and intestine, but mainly GGT is detected in serum that is derived from the liver. Hence, GGT acts as an important biomarker of hepatobiliary disease. Due to toxic or infectious hepatitis, there is a moderate elevation in GGT that could be around 2 or 5-fold increase. So in cholestasis, that is called, which is nothing but this gallstone formation, intrahepatic biliary blockage leads to 5 to 30 times higher serum GGT than the normal level. Yes, serum GGT can also be elevated in other diseases conditions such as hypothyroidism, rheumatoid arthritis, myotonic dystrophy, and OPD, obstructive pulmonary disease. Here, the GGT to platelet ratio index presents a novel marker of liver fibrosis in patients with chronic hepatitis. So the research is going on, being conducted to evaluate the use of GGT as a successful biomarker for nephrotoxicity and cardiovascular disease tests. So far we have seen liver associated enzymes to diagnose the diseases that happens in hepatic regions. Now the other prominent area of enzymes used in diagnosis is in cancer. So what do you define as a cancer? So cancer can be defined as a disease in which groups of cells grow abnormally, resulting in their uncontrolled growth or proliferation. So this proliferation can be fatal if it is allowed to continue and spread, which becomes malignant. Moreover, 90% of cancer related deaths are due to the process called metastasis, that is benign to malignancy. So it occurs in many different body parts including lungs, breast, colon, prostate, brain, mouth or even in the blood. So during the diseased condition, the level of certain enzymes in the bloodstream becomes abnormal, which further acts as a biomarker of cancer prognosis. A few enzymes that are involved in the detection of cancer are mentioned, which is given here. So acid phosphatases, cathepsin D, cysteine cathepsins, cyclooxygenase 2, dehydrogenases, sorbitol dehydrogenase. Next we are entering into another next subtopic, that is enzymatic biosensors as diagnostic tools. So what do you mean by biosensors? First of all, you should know what you mean by a biosensor. A biosensor is nothing but an analytical device that incorporates biological or biologically derived sensing material with close proximity to the physiochemical transducer. So it can be biological or biologically derived sensing material. Here enzymes are nothing but biological substances. So we can consider here, so enzymes as a biological material, and the analyte involved here can be both as a biological material as well as a biologically derived sensing material. Okay, so this diagram shown in this slide will tell you the explanation of the given definition. So this is an analytical device where your analytes are analyzed using this immobilized enzyme. From this immobilized enzyme, the signals are transduced in various ways. The prominent ways are amperometric, optical, thermistor and piezoelectric. So where it is converted, the signals are converted into digital signals and then it is amplified and shown in microelectronic devices. So the main purpose of such a device is to generate a discrete or continuous signal. So the signals generated from here are either discrete or continuous signal which is proportional to the concentration of an analyte. So depending on the concentration of analyte here bound to this immobilized enzyme, you will get the signals. So which was the first biosensor that was discovered? So it was developed in 1962 for the detection of glucose, that is glucose based biosensor. So the enzyme involved to analyze this glucose is glucose oxidase. So the electrode which is used here is oxygen electrode, as a transducer. So oxygen electrode is used as a transducer. But then on, the concept of enzyme electrode has been popularized and implemented in various other enzyme-based biosensors. So later on, after this kind of that is enzyme-based biosensors using electrodes, there came later stages potentiometric urea biosensor, which is developed for the detection of urea in clinical samples. The biosensor was constructed by immobilizing urease. In case of glucose, it is glucose oxidase. Here for urea, it is urease. And the ammonium ion selective electrode as a transducer. So the electrode used here is ammonium ion. So here the transducing element is thermistor. What do you mean by a thermistor? It is a heat sensing element. So it will measure the heat generated from the conversion of urea by this urease. So it has been utilized for the development of a biosensor based on a thermal enzyme probe. And then comes the category of optode, which is optical based transducing elements. This was discovered by Lober and Opitz. They have only coined this term, optodes. This consists of a fiber optic sensor with an immobilized indicator to measure carbon dioxide or oxygen. They extended this concept to develop an optical biosensor for alcohol by immobilizing alcohol oxidase on the end of a fiber optic oxygen sensor. So here are the list of electrodes or any other transducing elements that are commonly used as biosensors. So this table will give you an understanding of these different elements used in biosensors. But we are merely concentrating on medical terminologies as well as concepts. So I am not going into technical details about these biosensors. In the before slide, we have seen the categories of biosensors based on the transducing elements, how they are categorized. In this slide, we are going to see how biosensors are classified based on the discovery in their years. So it is divided into first generation, second generation, and third generation. But these examples are based on amperometric biosensors because those are the biosensors that was first discovered. So amperometric biosensors have been divided into three generations. On what basis? Based on the working principle. Okay. First one we will see the first generation biosensors were projected by Clark and Lyons, and then it was implemented by Updike and Hicks, who denoted the term enzyme electrode. So the enzyme electrode described by them was comprised of an oxidase enzyme. So now, until now we are seeing that glucose oxidase, which is immobilized onto a dialysis membrane on a platinum electrode. Okay, so platinum electrode. So here they have told immobilized enzyme. So consider this as a glucose oxidase. It is implanted on a electrode known as platinum electrode. So and it is a dialysis membrane. So what it will record? The depletion of O2 or the formation of H2O2 is subsequently measured by this platinum electrode. So what it will measure? So it will measure either depletion of O2, so which is being converted, it is the reactant that is converted, and the product here formed is H2O2, that is formation of hydrogen peroxide. So these two elements are measured by that platinum electrode, and which is the catalyst that is converting glucose oxidase. So this is recording the first generation biosensor. Next, second generation biosensors. The second generation biosensors have been commercially mostly in a one-time use testing platform. So it is a one-time, it is like use and throw platform. So the first company that marketed this second generation biosensors is Medicines. So it is based on Massachusetts in United States. They have launched this second generation biosensors as a product. Regarding its application, it is used in blood glucose monitoring, but this device was only for home use. Its mediation was provided by ferrocene species, that is this iron containing species, which is labeled here as Me. Next, the last category, the third generation biosensors. It is marked by the progression from the use of freely diffusing mediator to a system. So freely diffusing mediator to system. We can see the shift of immobilized enzyme shifted from here to above the transducer. Before, the transducer and immobilized enzyme, they are separated, but here you can see there is a shift. So that is the progression. So freely diffusing mediator to a system where the enzyme and mediator are co-immobilized. So the immobilized enzyme and the mediator are co-immobilized. That is, for example, you can take platinum, are co-immobilized at an electrode surface, making the biorecognition component an integral part of the electrode transducer. So it gives an integral component where there is no entropy involved here regarding the escapism of these substrates or products. So that's how third generation biosensors came in place in this discovery. The co-immobilization of the enzyme and mediator can be accomplished by redox mediator. So reduction oxidation mediator, labeling of the enzyme. So this enzyme is labeled by a redox mediator, followed by enzyme immobilization in a redox polymer, or enzyme and mediator immobilization in a conducting polymer. So in this polymer it is immobilized. So there are various biosensors that have been developed into diagnostic information. So one such examples can be given in here. For example, glucose dehydrogenase, cellobiose dehydrogenase, horseradish peroxidase. So these are the prominent examples of third generation biosensors. So in the previous slides, we have seen biosensors based on the technical efficiency as well as in their upgrading of the technologies, but strictly in the devices category. But now we are going to see this application of biosensor in a category where this biological mechanism of enzymes are being updated. So one such category is allosteric enzyme based biosensors, where this enzyme is updated here. So which has a novel mechanism which is known as allosteric enzymes. So you have to know what is allosteric enzyme. So what do you mean by allosteric enzyme? So what does it do? It exhibits regulatable catalytic activities. That is regulatable catalytic activities. For a normal enzyme, it has catalytic activities but regulated by another external mediator. But in case of allosteric enzymes, allosteric, so it is self-regulatable due to their steric regulation. Steric regulation means their three-dimensional structure controls from one place to another place. So we will see it. So it exhibits regulatable catalytic activities upon the binding of an effector molecule to a receptor site of the enzyme, which is different from the active site. So the receptor of the site of the enzyme is different from the active site. Normally in enzyme, these two sites are placed in one place or nearby to each other. In some cases, modulation occurs through binding to distinct alternative sites, either inhibitory or stimulatory. So as I am telling you now, this allosteric mechanism takes place in a different place where these two areas, where this active site and the binding site, they are in distinct places. That means it will be present in various orientations in various places of an enzyme structure. So it has two roles, either it can be inhibitory or stimulatory. So it catalyzes the formation of easily detectable products as potential biosensors. So now these biosensors can be more potentiable if these kind of enzymes are placed in here, that is allosteric enzymes. So normal enzymes, they have the catalytic role but they don't have prominent regulatory role. But if you consider allosteric enzymes in biosensors, it plays catalytic role as well as regulatory role, and in same way either can be stimulatory or inhibitory. So you are having here a multipotent enzyme based biosensor, that is allosteric enzyme based biosensors. So in this diagram it is self explainable, that you can see that So now catalysis or catalysis depending on the allosteric enzyme. So substrate is being converted into product in this site, but the effector molecules, either it can be stimulatory or inhibitory, involves here. So it is distinctly placed. This is how allosteric enzymes are achieve their mechanism in this way. So regarding different allosteric enzymes as biosensors, this table gives you a overall picture of various allosteric enzyme based biosensors. Okay, for example, we can take this beta galactosidase. It catalyzes the, what is the function of beta galactosidase? It catalyzes the hydrolysis of lactose to glucose and galactose. So lactose is converted into glucose and galactose. For this type, they are using optical transducer biosensor. And here, for this product conversion, there are substrate analogs are present here, which is nothing but XGAL and ONPG. XGAL is nothing but, what is its scientific name? It is 5-bromo-4-chloro-3-indoxyl L&D galactopyronoside. So this is the technical name of XGAL, and ONPG is nothing but ortho-nitrophenyl LD galactopyronoside. So they have strongly supported the use of enzyme as a reporter for the detection of gene expression. Now these are the recent discovery of enzymes as biosensors using allosteric enzymes, where beta galactoside instead of using normal substrate, they are using analogs where XGAL or ONPG used. So they support what they support. They are support as a reporter molecule for the detection of gene expression. Okay, so for detection of gene expression, which are used in antibodies for the diagnosis of diseases like HIV or hepatitis C. So here the effector molecules are antibodies. So antibodies which will recognize those peptides, especially if you are taking example for HIV, it displays an antigenic peptide. So that is recognized by these antibodies, and these antibodies are recognized by these analogs, which is indicated by their chromogenic substrates converted by this beta galactosidase. So that antigenic peptide, it is present in the, for example, what is the antigenic peptide present in HIV? It is nothing but a GP41 structural protein. So glycoprotein 41. Okay, which gives you an excellent response against human serum. So if you take the sample of human serum and if you want to test this antigenic peptide, that is GP41, this beta galactosidase allosteric enzyme biosensor will give you a best indicator to identify that, where XGAL and ONPG are used as chromogenic substrates instead of normal substrates. And the last category of enzymes as diagnostics, which are nothing but commercially available enzyme-based biosensors, which are self-diagnosable. As in the first slide, I have told you in the introductory slide, they are self-diagnosable. So here are the examples I am going to give you regarding those commercially available enzyme-based biosensors, which are self-diagnosable in the home itself. So at present, there are more than 500 companies worldwide that are working in the field of biosensors as well as bioelectronics. We can call this commercially available enzyme-based biosensors as bioelectronics. Okay, so one such promising company which have revolutionized in this field is Yellow Springs Instrumental Company from Ohio in the United States. So they are giving practical applications of these biosensors. Their first commercial biosensor is glucose biosensor, which was launched in 1975. It was based on the electrochemical detection of hydrogen peroxide, and the enzyme used for the development of biosensor was glucose oxidase. So that first biosensor, which I have explained before, glucose oxidase biosensor, it was discovered by Yellow Springs Instrument Company. Later, Clemens et al. Introduced a new amperometric glucose biosensor. Maybe you can tell it as second generation biosensor. It is a bedside artificial pancreas. It was marketed by Miles under the brand name Biostat. Regarding the term bedside artificial pancreas, it mimics the normal functioning of pancreas, where it can just regulates the level of glucose bound on it, and tells you the result. So that's why it is known as bedside artificial pancreas for this amperometric glucose biosensor. And then moving on to more such discoveries, like in 1987, other types of electrode materials such as screen printed enzyme electrodes. So screen printed enzyme electrodes are nothing but a paper strip like elements where results are printed in the screen itself. Such kind of technology is evolved in 1987. So, and other such promising discoveries are uh, given such as like cholesterol oxidase biosensor, which is marketed as from Livestream Technologies, and from Kyoto, Japan, it is marketed as Autostat. So these are the commercially available enzyme-based biosensors that came later into the market. Now, in the latest discovery that goes on, now recently they have discovered optical visualization and quantification of enzyme activity using dynamic droplet lenses. So, using dynamic droplet lenses, they have discovered modern technology, and it is based on smartphone devices. So, they have discovered using smartphone also can be used as a biosensor. So, this is gives you the emergence of technologies where you can use smartphone driven personal health monitoring, and your smartphone acts as a point of care devices for this kind of biosensors. So here the technology used is, what is the technology? Dynamic droplet lenses, where the droplet lenses, where the and you, if you are using the smartphones, you can see that there is a sensor, light sensor, that is placed on your mobile phone. That line sensor will detect your sample sunlight and will tell you the proximate result. So this is the modern research that is being commercialized for commercial biosensors. So to conclude, so biosensors have the prominent role in each and every of disease recognition as well as a biomarker. So the most successful commercialized enzymatic biosensor is the glucose biosensor. So that is what we have seen in many examples and many subcategories. So glucose biosensor is the most commercialized enzymatic biosensor for used for the detection of glucose levels in blood samples, because diabetes is a not curable disease. You can just elevate or decrease the amounts of sugar levels in your blood. So it is, so glucose oxidase biosensor is a most commercialized enzymatic biosensor for diabetes, for everyday use for diabetic patients. So with the advances in this kind of enzyme technology, there is no doubt that enzymes and enzyme-based biosensors will become a powerful tool. Even it is being now, it is the research is going on to test for other diseases such as cancer also, and for heart failure, epilepsy, and many more other such kind of diseases in the near future. So now it is being used on a laboratory scale. However, there are many complications during the analysis of real samples. So now, likewise, I have said that it can be used in smartphones also, but it can be used for only preliminary analysis. You cannot take it as a guaranteed result for your disease indication. So it gives you a preliminary analysis, and it is only in the laboratory level. So where does this kind of technology leads to? So it is we have to develop novel surface modification strategies into the enzyme electrode in order to eliminate the non-specific adsorption at the surfaces of the enzyme electrode. So non-specific adsorption. So the layer which you are using as a polymer between the transducing element and immobilized enzyme surface, it has to be a novel surface that should avoid non-specific adsorption, and it should involve a large number of sample analysis. So diagnosis of large number of samples, where it should have the capability of multiplexing. What do you mean by multiplexing? At the same time, it should analyze numerous amount of samples in a single platform. It ultimately saves you the assay time, and it can be developed into a microarray also. We have seen microarray, right? DNA microarray, protein microarrays. So it can be developed into a microarray also, so which will also help you to save the assay time. So which performs the analysis of large number of samples, and it is also important, utmost importance in clinical diagnosis. So with this, it can be converted into, if these technologies are taking place, even then it can be considered as a lab on a chip device also, LOC device. What do you mean by lab chip device? So constructing them into a miniaturized platforms as enzymatic biosensors. So they can be called constructed as miniaturized enzymatic biosensors using thermostable enzymes, which will increase its portability and can be used as LOC device, that is lab on a chip device, or a point of care devices. Point of care devices means mobile devices. Then and there you can take it and use it as a device. That is called a point of care devices. And regarding biotechnology, so recombinant enzymes are also utilized in this lab on chip techniques for enzymes as diagnostics. So enzymes as well as enzyme based biosensors can be applied for the detection of various very low level targets within an appropriate time. So if you are using these recombinant enzymes, you can increase that sensitivity so that they can detect very low level targets within an appropriate time. So, until now we have discussed all the areas of enzyme based biosensors, only highlighting a overall view of all those topics. So, with this I am going to end this topic, enzyme based biosensors. Thank you.